All right, so we're going to go on and start with section 7.4, which is going to deal with gene regulation. So some genes are expressed the entire time an organism is alive. Some are turned on for short stints and then turned back off or maybe turned back on. Some are not on during young development stages, but are on later in life and vice versa. So what controls this? What makes genes turn on and off, so to speak? So gene is expressed when it is transcribed into RNA and then into a polypeptide, which is a protein. So only a fraction of the genes are going to be expressed at once. Okay, again, some are on, some are off. You may even carry some genes that are never ever turned on, but they may be passed on to the next generation. So a cell regulates the genes which are expressed in order to save energy and control the cell process. So again, remember and think back to um, go ahead and think back to how proteins control everything. So in prokaryotes, it's a little more simple because they're simplistic cells. Prokaryotes control the expression of genes by using mechanisms called operons. Operons are groups of genes that regulate gene expression. So simple operons are made up of a repressor and an operator. A repressor is a molecule that stops RNA polymerase from binding to the promoter. So you have to remember what RNA polymerase is. An operator is a section of the gene that the repressor binds to. So an example of this would be lac operon. So this is a molecule that's going to break down lactose. So the repressor is bound to the operator, preventing transcription, step one. Step two, the cell takes in lactose, which requires a special set of proteins in order to break it down. Step three, lactose binds to a repressor and the repressor releases from the operator. Step four, enzymes that break down lactose are made and they break down all the lactose in the cell, ending with a molecule of lactose that is bound to the repressor. Step five, when lactose is no longer present, the repressor binds again, it should say to the operator, preventing transcription. So this is the generalized, this is one specific example of how prokaryotic cells will regulate certain things as far as genes being expressed. So in eukaryotes, it's more complex cells, so the process, as a result, is going to be more complex where there's more genes and there's more cell differentiation, which takes us into cell differentiation. So we've talked about cell differentiation in the past. Cell differentiation takes on this is when cells take on different roles as the organism develops. Stem cells are cells that have a blank slate, so to speak, and the ability to become any kind of cell. Stem cells are found in early stages of animal development, as well as certain tissues that are continually creating new cells. So, even say in you right now, bone marrow is an example of stem cells. Okay, new cells are being produced right now within your body. All right, so Hox genes. Hox genes, control differentiation in multicellular organisms. Hox genes can control the position and location of different parts of the body. Hox genes are active during development and are turned off once the organism is born. Other genes are regulated similar to Hox genes, and they are no longer produced after the initial embryo development or are only turned on at certain times. An example of this is tissue repair. All right, so the gene that regulates your repairing skin from a cut is not going to be activated and turned on unless there's a reason for it. So if you don't have a cut, then this gene won't be turned on and be turned off. All right, so cell signaling. We talked about cell signaling when we talked about the nervous system. This is communication between cells. Cells receive chemical signals from the environment, from each other, and from the environment. Differentiation can occur as cells communicate, detailing information about the position or location. And so basically what this means is signals and information taken in from the environment around a cell can cause certain genes to become active or certain genes to be turned off. So this is another example of cell differentiation. So relatively short presentation this time. Make sure you complete the form that goes with this. And if you have any questions, please let me know. And we're getting ready for the first test of the unit. There's 2 tests in this unit, and the first one will end with 7.4. So if you have questions, let me know. Thank you.